All right, yeah. So as a reminder, if this is not relevant to you, feel free to keep working here or work in the other room or elsewhere in the building. Yeah, this talk is focused on how to convert data to NWB because this is relevant to a lot of experimentalists and maybe other folks in your lab who might be interested in this. So as I mentioned in the first talk, converting data to NWB presents a number of challenges. There's many different formats. Many experiments require integrating multiple data sources together with their own formats. SpikeGLX, deep lab cuts, the raw, this processed data. And these data are going from a few 100 megabytes to gigabytes to maybe even a couple terabytes together. And if you have data like this, you might not know how to approach it because it's very challenging to figure out how to convert all these data together, align them, load them into your computer such that you don't overload your RAM and crash your computer. And how do you understand the standard on top of all that of organizing the data such that it can be reused and uploaded to Dandy. Right, so we present, to help with this problem, we present multiple ways of converting your data to Dandy, to NWB, and then uploading to Dandy. You can use NWB Guide. This is our no-code automated conversion app. Presents a graphical user interface, walks you through step by step, and I'll demo that in just a minute. This works for many input data formats, but not all of the ones that are out there. For a larger set and more flexibility, we have NeuroConv. This is a tool that presents a low-code solution and requires you to write a handful of lines of Python code. It's not too bad. And if you have custom formats that are used in your lab, custom NumPy files or MATLAB or Excel files, or you're working with data that isn't supported by the NWB standard, you can use our core NWB APIs, PyNWB for Python, MatNWB for MATLAB, and some of you are already using these for reading data. You can also use it for writing data. Okay, so then I will demo guide. So to find guide, just Google NWB guide. Hopefully one of the first results is the NWB guide documentation. Just go here. Install. We provide installers for the different operating systems. So I did this about 5 minutes ago, downloaded the Mac installer, and then ran the installation. So now I have guide here. Hopefully that's big enough, yeah. Okay, so we're just gonna go through a very simple conversion here. Click start. We're gonna say, this is just a test. Next. For this example, we're just going to convert one single session from one single subject, and guide will ask you details, metadata about your experiments that's is almost never available in the source data, but is useful for storing in the NWB file and sharing with others. So we'll just say the subject ID is sub1, session ID is session1. We'll turn off publishing data on Dandy for now. And you also have the option of customizing low-level data storage options, so customizing your compression or chunking. But we provide great defaults that should work for everyone. Okay, then you go here and select the data formats that you work with. If you scroll through this list, you'll see a number of formats that may be familiar to you. There's alpha omega for extracellular EFIS, BlackRock, Bruker imaging for calcium imaging, Kaiman segmentation, if you use the Cell Explorer tool, deep lab cuts, extract for cell segmentation, kilosorts, miniscope data, Neuralinks, etc. Yeah, the list goes on. We've put in a lot of work to support all of these different formats. And for this demo, we're just going to use a SpikeGLX recording. SpikeGLX is commonly used to acquire NeuroPixels data. And we'll add sorting from the Phi spike sorting curation tool. Okay, next. Now Guide will ask you where all this data is. So let's go to Phi, and we have some test data that I generated earlier. And I'm gonna go and navigate to that. So test data, single session, Phi, and Guide is asking for the folder with all of the Phi NumPy files and associated files in it. So let's say that. And for SpikeGLX, it asks for the ap.bin or lf.bin file, so the high frequency band file or the low frequency band file. There's also a separate option where you can convert all of the different streams that you have, but for this demo, we'll just select one of them. Okay, hit next. Now Guide will ask you for metadata about the experiment. So you can enter general metadata about who's doing the experiment or who did the experiment. Let's say this is from Janelia Research Campus and the Voights Lab. You can add additional information about how the data was collected, description about the experiment. You can add an experimenter, multiple experimenters, keywords that will help for search on the Dandy archive, any notes about the experiment, the pharmacology, the protocol, publications, etc. These are almost all optional fields, and if it was required, you would see a star next to it, and it would be red. And recommended fields are in yellow here, so I'll just add a description here. This was an awesome experiment. Okay, keywords, sure, I'll add one. Neurophysiology, sure. Okay, and then we can expand the other sections. So subject, we'll select the sex of the animal. Let's say we're working with a female mouse. And we have to specify the age. The age goes in a specific format, starts with P, and then a number and a letter code for the unit. So we could say P90D for 90 days, or P1 year and 10 days. We'll just do that. And you can add additional information about your subject, description, genotype, strain, weights, etc. But we'll leave those empty for now. Okay, then Guide will do a preview conversion. So this will convert just a small subset of your raw data. Your raw data might be 100 gigabytes. That's actually pretty common. But Guide will just subsample that to a few channels and a few time points and run the conversion, make sure so that it can do a few validation checks and present you what would be a stub of your data so you can review it to make sure it looks good before it spends the hour or so converting the full data. So the inspector has scanned your files and checking it for adherence to best practices. So you'll see here there's a few missing recommended fields, a description for the subject. These are descriptions of some of the columns that have been extracted from the Phi data that we could have edited. And none of these are errors, so we'll just move on. You can go back and correct these if you'd like. And then Guide will open a embedded NeuroSift view of this data, and you can full screen it and verify that the metadata is entered correctly on the left here, and also within the file. So you can open up your electrical series and look at it and make sure the time axis is correct, the channels are correct. Here we have 384 channels of generated spiking. And it should only be, I think, a couple seconds, 3 seconds. So that's all that was converted in this test. But it's important to visualize your data, make sure it's all correct during the conversion process. You can also look at the electrode information that was extracted from SpikeGLX and Phi. That's all there. And the units you can look at as well and plot rasters, even though you don't have all the full data, but just to make sure this looks correct. When you're happy, we exit full screen, and we'll hit run to run the full conversion on the full data set, 100 gigabytes. And this can take some time because we're compressing and reorganizing your data to optimize it for storage, especially on Dandy. All right, done. You can click this to open up your OS Explorer to see where your NWB file is, move it around, etc. And Guide will convert most of your raw data, great, but sometimes it doesn't know how to convert some specific data that you have, or you want to do something special. Well, you can use Guide to start your conversion, convert as much data as you can, and then you can use PyNWB or MatNWB to open the file that you've generated with Guide and append additional data that is missing. So one thing that we don't have support for yet in Guide is the trials table. And so you can go in, open up the NWB, open the file with PyNWB in append mode here, and add your trial information. And we provide some guides down here. Okay, so this was a single session, and then I'll briefly show you what we do to do batch conversions. You wouldn't want to do this process for all 100 sessions of your experiments. So I'll show you how we do that very briefly. You select yes here, and most people have organized their source data from all 100 sessions in some systematic way on their computers. And so we can tell Guide to locate this source data programmatically. And let's say all of this source data lives under a root folder. We'll just say it's under there. Okay, and we'll publish on Dandy. Then we'll go and select SpikeGLX again, this one, and Phi. And Guide needs to find your data. And so we'll select the base directory. Has already been selected before. You can customize it. And then we'll go to this autocomplete feature. And Guide asks you to select one example folder of a Phi recording or data from a single session. So we'll go into multi-session data sets, which is our roots. We'll just select under mouse 2. Let's say we organized our data in this way, where we have the top level directory is the subject name, and then under that, it's subject name _ session, and under that are the individual data streams. And this might be different on different people's computers, but Guide is flexible in supporting variations. So let's say we select mouse2 _ session1 _ Phi, hit open, and then Guide asks you, all right, given this path string, what is the subject ID and what is the session ID? And so we can say mouse2 and session1. If you wrote mouse2 in session 10 or session 1 point something, or some string that isn't actually in the path, Guide will complain and ask you to fix it. And you might need to reorganize your data to make this work a little better in your system, but in testing we found that this works for most lab setups. Hit submit, and Guide will have searched through this root directory here to find all of the matching source files in your that correspond to Phi sortings in your on your computer. So it found 4 source files, and we'll do the same for SpikeGLX. This time we'll select the ap.bin file for mouse 2 session 1, and we'll do the same. And again, it found 4 source files. Great. And now we have a table to enter the metadata for each subject instead of the individual display. It'll ask you, all right, what is the sex for mouse 2? Let's say it's male, mouse 1, female. Let's say we are working with Drosophila in this case, and I don't know, a human in this case. And let's see. 10 days and 30 days old, sure. You can also edit default metadata on right here. Who knows? I mean, you might have an experiment that has a fly and a human. Yes. Unfortunately, Guide isn't smart enough to find any unintentional mistakes like that. You can also edit metadata on this form that will apply to all of your subjects. Let's say you have 20 subjects, you don't want to have to type that they're all mouse, you can do it here. Or that they all have the same genotype or something. You can apply it here and it'll auto-populate down the field. All right, then here's the source data. You can click through the accordion, the expansion things, and confirm that the Guide has found the right data for the right sessions. There's two mice, 2 sessions, or one fly and 1 human. And then you have to specify any additional file metadata. So this was extracted from the table. You have a male fly as mouse 2. And you could also add information about the institution, the lab, and all these additional things. So mouse 1 data might have been acquired from experimenter A, and mouse 2 might have been acquired from someone else. And you can specify differences per subject or even per session here. If there's something that applies to all sessions, you can go into edit default metadata and supply it here, and it'll populate across everything. Great. So then you hit next, and Guide will extract a stub or like a small sample of each of these files and run a conversion. So we have 4 sessions, 4 files, and just like before, you can inspect them for any for adherence to best practices, go back and correct anything. This is the report for all files, but you can go into individual subjects and sessions as well. When you're happy with that, you can go visualize your data, just the same, navigate through each subject in each session. And when you're happy with that, hit run, and this will batch convert all of your data that you've selected for all of your sessions. So this could be 20 subjects, 10 sessions each, 200 sessions. This could take, I don't know, probably overnight at this point. And then you'll have all of your NWB files. And if you have nothing else you want to add, or even if you do have things you want to add, you can go into PyNWB or NWB, make your additions, and then come back here. You can come back to this pipeline. You can hit next. And to initiate the Dandy upload. This we find is the easiest way to upload your data to Dandy. It asks you for an API key. Normally, you would go to the Dandy archive and click up here, and your API key is this little hidden thing over here. Copy that and paste it here. For testing, I'm going to go to the test archive, which I never remember the URL because it changes. Okay, so I already have an account here, but if you don't, I think you would have to log in with GitHub here. And I'll paste that here. Good. Can you explain the sandbox? Oh, yes. So if you are running through this tutorial, which is on NWB Guide, or you want to just do a test upload to Dandy before making anything public, or you're just testing how everything works together, we highly recommend that you use the sandbox. the sandbox archive. This is a separate archive from the main archive. The data here are not guaranteed to stay there forever, and might be just deleted randomly. But this is where you can test how Dandy works, how editing the metadata works, how downloading works, before you submit to the main archive. And we have this so that the main archive isn't polluted with test data that isn't real and isn't useful for anyone. Okay, so specify your API key, hit submit. Yeah. And now we can create a new Dandy set, provide a title, test at Janelia. Say we're submitting to the development server, test guide, upload. And then here you'll have the option to select a license. I think as we mentioned earlier, there you have two options, CC BY and CC 0. And Dandy has a handbook that can help you select which license is most appropriate for you. And then you also have the option of embargoing your data sets, which will keep it hidden from public access until a specific time period. We'll just say no for now. Okay, hit create, and this will create a Dandy set with number 217046. You can customize the upload settings, but once you're ready, just hit upload. And the 4 NWB files that are part of this conversion pipeline will be uploaded to Dandy. Oh, and this is usually fast. Okay, great. Success, it's there. We can click this, and we can see our test data sets, 76 megabytes, 4 files. You can go into the files and see that we have all our files here. You can see that I am an owner of this Dandy sets. I can delete files here, and I can also go back and go to the metadata and edit the metadata related to this Dandy set, providing additional information that will help guide folks searching for related data. And when I'm ready and everything is validated, I can hit publish, and this will mint a DOI for the Dandy sets. Oh, I can do it now. And fix it in time. And then you can publish new versions if you make any changes. Again, for the tutorial or for any test data, make sure you use the sandbox. You can see I have at least 30 test data sets on the sandbox. And this would not be good to have on the main archive. Okay, so that's the main conversion pipeline. Guide also provides features for you to standalone features options to validate your data. Just drag and drop or select an NWB file here, and it will validate it for you. You can also drag and drop an NWB file and explore it. Let's see, I just downloaded this 517 gigabyte NWB file. I'm just going to drag it in, and this will launch NeuroSift within Guide, and you can full screen it and do all the things that you would do on the website, but this data hasn't been, let's assume this data hasn't been published yet, and you want to spot check it or explore it. And it also has a standalone feature for upload where you can drag and drop in there and create new Dandy sets or select an existing Dandy set to upload to. Okay, any questions on Guide?
It said in the embargo piece that you needed to have an NIH award number to be able to embargo?
Yes.
Is that true?
I think if you are not supported by an NIH grant, I think the advice is to provide some information about your grants, assuming you're funded by grants.
It's a little bit awkward with the forms, because the forms kind of assume NIH funding, but our policy does not constrain that.
Yeah.
So you just kind of have to work with it a little bit.
We like the idea of being attached to some piece of funding, which...
Right. Yeah. I mean, if the forms don't allow you to do that, just document it clearly.
And then if you
So it depends on the problem, but if the
The data itself.
the data itself, then let's see, tests at Janelia. Okay. So that means all of these files are invalidated, or you want to replace them, right?
Let's say either one or all of them.
Okay. Then you would delete the files that you don't want, add new files. It's not necessarily a re-upload everything from scratch, as if it's a blank slate. You start from your current state.
And then if there's a problem with everything, what would you recommend doing? Hypothetical.
So you could do Dandy download on the Dandy set, make all your changes locally, re-upload, and I forget if there's like a force option or like a replace option.
Yeah, I've used the Dandy CLI. It has tools for like programmatically managing these assets. Like if you have 100 assets and you would need to replace all of them, I wouldn't go through and
Yeah.
well, 100 maybe you could, but yeah, you get the point. The CLI is built for that.
Yeah.
So then the guide, you probably want to do something like hybrid between the CLI and the guide.
Yeah.
Okay, cool. And you would recommend doing that instead of deleting the whole Dandy set and starting
I
again?
think if what you're talking about are corrections or additions, then it should be a new version of the same. If it's the same
new version of
underlying
the same
experiment,
Yeah. Yeah. Keep the lineage and yeah. Okay. Any other questions on this part, on just guide? All right, so just to recap, guide provides a simple interface, converting data, supports 40 plus formats, enter metadata, and it's validated on demand or immediately, and you can get the documentation. Use NeuroSift to visualize it, and batch convert everything, and then you can upload directly to Dandy. If guide doesn't work for you, for whatever reason, there are quirks with different operating systems and requirements that some formats don't work on some systems. You might be able to use NeuroConv, and we recommend you try this next. With only, I don't know, this is less than 10 lines of code here, you can build a Spike GLX converter, and where you identify, or you tell NeuroConv where is the data, adjust any metadata. Here we're setting the session start time, and then run the conversion, just calling converter.run conversion. And you basically swap out the name of the converter here with the corresponding converter for any of these data types. In addition, you can also use NeuroConv to combine data from multiple file formats, just like in guide. So your Spike GLX, and then you can add a PHY conversion or a Suite2P, imaging, etc. And so here's a list of the 45 plus formats that NeuroConv supports. I think a few more have been added on this list since I made this slide, but you can kind of look through and see if yours is on here. And if it's not, we ask that you want to use NeuroConv or guide, we ask that you submit an issue ticket on the NeuroConv GitHub repo telling us what is the format, and if there's any small sample data that you can provide, it would help us a lot in building out support for automatically converting that data into NWB. Okay. And if neither guide nor NeuroConv work for your use case, you're working with extensions, you are working with custom trials, or you want to really optimize or customize how your data is compressed, you can use PyNWB and MatNWB, and we provide a ton of tutorials that are domain specific. So extracellular electrophysiology, how do you write the electrodes table and the electrical series and your units. We have tutorials on optical physiology and intracellular electrophysiology, behavior data, in both PyNWB and MatNWB. And those are just on our docs page here. All right. Yep, so just to recap, converting data to NWB can be daunting, but we provide these the 3 sets of awesome tools to help you through the process. NWB Guide, it's the easiest. You don't have to write a single line of code if it works well for you. Just click through, select your data formats, enter your metadata, and your data will be converted and uploaded to Dandy. NeuroConv for a little more fine-grained control and a wider scope, and PyNWB and MatNWB is the most flexible, lets you do basically anything to convert your data to NWB. Yeah, all right, that's it. Any questions? Okay. We also have host one-on-one consultation sessions. So if you are having trouble converting data to NWB, please reach out. We can set up a Zoom session or to talk over your specific use cases. You can also post on our GitHub repos for PyNWB or MatNWB or the NWB help desk for if you don't know where to ask discussions. And we already have a growing list of discussions here yeah. All right. Steph, Ben, anything I missed? Okay. Well, I'm here to help. So let us know if you need any help with this now or in the future. And back to hacking.